transnational companies like Pepsi, Kraft, Nestle, Campbell's. And what they've been doing is that they grabbed kidney cells from human aborted fetuses. So they grab these kidney cell lines, las células sanguíneas de los riñones de bebés abortados, and in laboratory they replicate these cells and they put them in all your products. So ponen los productos. What products are we talking about? Well, we're talking about your Gatorade. Drink some aborted babies. Yes, seriously. We're talking about Lay's chips. Yeah, that's why they taste so good, don't they? Pepsi products. All right? Tropicana juices, Dasani water, Minute Maid. We have to not consume anything from these companies. And tell them that we're not participating in their insane ritual that creates.
Read about it, questions and answers. Read about it, questions and answers about nutrients. Volume 4, Dr. Phyllis B. Canyon, Sons and X boosters. We gonna get into this. The boys stay on this shit. Like, come on now. I might just read this shit myself, man. I was trying to be lazy. I really was. Okay, there we go. Yeah, they allow me to be lazy.
Dr. Phyllis B. Camion. In small quantities so that they fly under the radar for labeling. Because they are not actually ingredients, but are referred to only as flavor enhancers, they are not required to be listed as a product ingredient except as an artificial flavor. According to Joseph Mercola, MD, Cenomix boasts that they have discovered taste receptors using HEK293, which is human embryonic kidney cells taken from an electively aborted fetus. While HEK293 was originally used by Dutch scientist Dr. van der Ebb, HEK293 cells are becoming more widely used outside medical research. When news broke of this being used as a flavor enhancer, one major company immediately stopped using the product, although there are others that continue at this date. For a food manufacturer to advertise that their product contains 50% less sugar or to be able to greatly reduce the amount of MSG, monosodium glutamate, by using Cenomix but still have that sweet, savory or salty taste is indeed a marketing coup. Just as a footnote, Cenomix has not undergone the Food and Drug Administration's usual safety approval process for food additives. Rather, it gained the generally regarded as safe, GRA, status from the Flavor and Extract Manufacturers Association, an industry-funded organization, in less than 18 months. Unfortunately, the manufacturer's #1 goal is profit, not our health. Q. I have been asthmatic for a few years and was wondering if there are any foods that can help asthma? I eat pretty healthy, have eliminated milk and caffeine and drink half my body weight in ounces of water daily as you recommend and have noticed a big difference. Any suggestions would be appreciated. HEK293 protocols: cell culture, transfection, protein production. HEK293 cells are popular for their ease of growth and transfection. HEK293 transfection kit, making them a common cell culture in cancer research. In addition, high transfection efficiency of HEK293 cells produces exogenous proteins or viruses for pharmaceutical and biomedical research purposes. HEK293 cells are useful for many transfection experiments, particularly the propagation of adenoviral-based and retroviral-based vectors.
Alright, now, none of us know what the fuck a transfection kit is, but we gonna find out, you know what I'm saying? Ain't no reason to be sitting here We came here to learn today. Let's do this. A transferrin cell line. Is it not what he said it was? Says the two components to the formulation enhance lipids, mediate transfection efficiency. The use transfection protocol provided for transfection of siRNA, DNA, mRNA, and microRNA. Kits include transfection enhancers, regent and recommend transfection protocol. High transfection efficiency is in the presence of serum. mRNA. Let's make sure we know what that is, because that's going on real big in that COVID shit. The messenger RNA is a single strand RNA molecule that is completely the one of the DNA strand of the gene that mRNA. The mRNA vaccine are not live vaccines, but do not use an infectious element, so they carry no risk of causing disease or vaccination in a vaccinated person. That ain't true. All right, so we got microRNA. A microRNA is abbreviation of miRNA, is a small RNA molecules containing about 22 nucleotides found in plants, animals, and some viruses. This function of RNA silencing a post-transcriptional regulation of gene expression. Main function are a class of non-coding RNA that play important roles in regulating gene expression. So basically how your genes are shown, like with how, like this thing, I guess would be like the motor, like the, all right, let's keep going, because I don't know, transfer DNA sequence into the primary mRNA that processes into a precursor of miRNA, and finally a mature miRNA. Yeah, and miRNAs may function as either oxygenes or tumor suppressors under certain conditions. This dysregulated miRNA have been shown to affect the hallmarks of cancer, including sustaining I have no idea what that is. Life-derivative signals, evading growth, suppressors, resisting cell death, activating invasion and metastasis, and including anagenesis. Although the precise mechanisms involved, the cadaverous biological functions are not known, and increasing numbers of studies suggest that mRNAs are important regulators of cadaverous growth and proliferation and cell differentiation and apoptosis. I have no idea. All right, let's keep it. All right, we're gonna find an experiment of this. siRNA is targeting laminin mcRNA or non-silencing controls. siRNA are transferred into HEK3 cells, 293 cells following the recommendation protocol at 48 hours post-transfection. The cells were analyzed by qRT-PCR for gene expression levels and 18 sRNA levels were used to normalize a laminin AC data values are normalized in untreated samples. Target species human for corresponding mouse products. See layman. Yeah, just some shit. Biological chemistry and mutant protein. Sharplanes, a rhesus mutant P53, a hyponic tumor, a hypoxic tumor. Okay. Yeah, let's not get lost in this shit. But we gonna. It seems like they're gonna show me a video. Y'all notice we want videos and we keep getting motherfucking articles. Okay. Vectorization of a rare human endeavorous. What does that mean? It's a common virus that causes a range of illness. It causes cold-like symptoms, fever, sore throat, bronchitis, pneumonia, diarrhea, and pink eye conjunctivitis. You can get endeavorous infection at any age. In a few days or a few weeks. Just sound like a common cold. There's no specific treatment. Endeavorous infections are mild and may require care and help relieve symptoms such as over-the-counter pain medicines. Usually spread through respiratory secretions when someone coughs or sneezes. But these stubborn viruses are tougher than cold and flu viruses. They can live for a long time on surfaces like doorknobs and they use many common disinfectants. They say you can buy stock in this virus. Oh shit. All right, let's keep going. HDV types and widen our knowledge of this family and their infections with cells and tissues and organs. In the study of HADV56, a member of the human AD specific D, and create ease of use of cloning systems to generate recombinant HD. Let's see what that is. This shit's starting to sound like COVID. Carrying foreign gene were present in vertical transduction profiles HADV56, a direct comparison of the most commonly used HADV5 base vector in vivo characterization demonstrates and deliver intravenously. All right, we're gonna keep moving because this shit is extra nerdy. Okay, we also investigated its potential as a vaccine vector. It says viral vector vaccines use live viruses to carry DNA into human cells. These DNA contain in our viruses encoding antigens that only expressed in infected human cells elect a immune response. It's triggered in recognizing foreign antigens in cellular context of activated dendritic cells with the expression of non-nucleotide cells. All right, we see they in it. Basically sending some foreign in there just to get your DNA to jump. The viral vectors vaccines combine many of the positive qualities of DNA vaccines with those of live vaccine. Okay, so the intended vaccine or a live or intended vaccine is a vaccine created by reducing the virulence of the pathogen, but still keeping it viable or alive, and leaving it dormant. Microorganisms. There are 4 types of COVID vaccine. Here's how they work. All right. You can get a whole virus. You can get a protein subnet. The structural biology of a protein subnet is a single protein molecule that assembles and co-assembles with other proteins and molecules in protein complex, some naturally occurring. Proteins have regularly small, or describing as ligament. I don't know. I can't fuck with it. I got a GED, a good enough diploma. Fuck with me. All right, we got the nucleic, any various complex or organic acid, such as DNA, RNA, they are composed of nucleotide chains. Okay, so you got a D, I guess. And then you got the viral vector. Viral vector vaccines are safe and effective. They use a modified, harmless virus to deliver instructions to our cells to trigger an immune response. All this is still tied into flavor enhancers. All right, existing products. It says there are no viral vector vaccines currently on the market for use in humans. There were 12 viral vector vaccines currently in use for veterinary diseases. The approved vaccines included antivirus fowlpox, which is chicken pox, right? I'm pretty sure. Fowlpox is a worldwide viral infection of chickens and turkeys. Nodular lesions and unfeathered skin are common in the cantuous form. Okay, yep, nasty. All right, yep. Let's keep it moving. All right, the antidate yellow fever or YFV17D. Massively parallels. Okay, wait a minute. Wait a minute. All right. Pause. So I find some long time for vacina virus vectors. The vaccinia virus engineers to express foreign genes are powerful vectors for production of recombinant proteins originating from high efficacious vaccines, securing worldwide eradication of smallpox. The most appealing use of vaccinia vectors is to serve as vaccine delivery systems for heterologous antigens, the development of modified vaccine viruses, anacara, and antigens. It says vaccinia virus infections is typically very mild and often does not cause symptoms in healthy individuals, although it may cause rash or fever. Immune response generated from the vaccinia virus infectious protectors these persons against lethal smallpox infection. So let's see what they. That was, let's get back to flavor shit. For some reason, it has a lot to do with this. So Says, please tell me which ingredient promoted you to allow this in your body. What is the aborted kidney cell, or maybe it was the aborted retinal cell, or maybe it is the citric acid you wanted instead of eating an orange, or possibly the ethanol, whatever prompted you. I am very interested in your answer. Please explain ignorance or maybe stupidity to me that put such a fear of dying that you would be a lab rat with things in it that you can not only be deadly on their own, but baby's life was taken in order to save yours. And you are okay with that? This version a little dark, but hey, keep it rocking. Do not argue with me or tell me that I am fabricating these truths. Read the ingredients, which, by the way, came from the FDA government site on every description came from the scientists. So please tell me again, how, God, is it okay with killing innocent children and have their cells, their DNA put in yours? Please tell me it does not matter if it was from a fetus murdered in 1975 or 1920 that was a baby in God's creation. You think I am being hard? You think I am being unfair? Okay, here we go. It's moving again. You think I am being unchristlike? Remember, he overturned tables after meeting a whip together to chase people out of the temple of God twice, and you are carrying the cells of a baby that was put to death because you feared dying from something that had a 1.1/10% death rate. People die in car accidents more than this. Let's get that off of them. You know, they out here dropping them jewels. So let's see what they talking about. Indigenous Carolina dropped a video today about aborted baby fetuses and food. Never did I get on top of, like, you know what I'm saying? Like, I never put it underneath the, you know what I'm saying, the Brian microscope, and I'm pretty pissed off. I'm a little hot. Coming in hot. What are the ingredients in Janison's COVID-19 vaccine? The Janssen COVID-19 vaccine includes the following ingredients: recombinant replication and competent and endovirus types, 26 expressions of the SARS-CoV-2 Spike protein, citric acid monohydrate, trisodium citrate, uh, dihydrate ethanol 2 hydroxypropanol B, xylodextrin HBCD, polyosobrate 80, sodium chloride, recombinant DNA genetic engineering, DNA molecules of DNA from two different species that are inserted into a host organism to produce a new genetic combinations that are of value to science, medicine, agriculture, and industry. Since the focus of all genetics is the gene, the fundamental goal of laboratory genetics is to isolate, characterize, and manipulate genes. Although it is relatively easy to isolate a sample of DNA from a collection of cells, finding the specific gene within the DNA sample can be compared to finding a needle in a haystack. Consider the fact that each human cell contains approximately 2 meters, 6 feet of DNA. Therefore, a small tissue sample will contain many kilometers of DNA. However, recombinant DNA technology has made it possible to isolate one gene or any other segment, enabling researchers to determine its nucleotide sequence, study its transcripts, mutate it in highly specific ways and reinsert the modified sequence into a living organism. What is recombinant DNA? Download a PDF copy. That old girl. But we already read that. We already went over it. What recombinant DNA was. It's cool that she gonna do that for us, but let's keep it pushing. Yolanda Smith. I didn't think that was a Yolanda. Let's go back to Yolanda one more time. Well, good goddamn, that is Yolanda. Let's back on out of there. Recombinant DNA, which is often shortened to rDNA, is artificially made DNA strands that is formed by the combination of two or more gene sequences. This new combination may or may not occur naturally, but is engineered specifically for a purpose to be used in one of the many applications of recombinant DNA. This article will go into further detail on what DNA is